Hello everyone, in this video we are going to learn what is electron transport chain or what is oxidative phosphorylation or how energy is produced on the inner membrane of mitochondria. As we know, mitochondria is a double membrane bounded organelle which has its own DNA and ribosomes. Both inner and outer membrane of mitochondria is a lipid bilayer structure, and inner membrane of mitochondria contains series of electron carriers that are called electron acceptors or complexes. The energy-rich compound NADH and FADH2 produced during glycolysis or Krebs cycle move toward the inner membrane of mitochondria. Here, energy is removed from the electrons and trap proton. Movement of proton from high energy to low energy gives ATP. Basically, inner membrane of mitochondria is a group of electron carriers. On the inner membrane of mitochondria, there are basically 4 complexes which are complex 1, 2, 3 and 4. Complex 1 contains 2 kinds of protein, that is FMN and FES. FMN is basically flavin mononucleotide and FES is an iron sulfur protein. FMN takes electron from NADH and transfer it into NAD positive and transfer the electron to FES2 positive, that is ferrous, and convert it into its reduced form, that is ferric, and transfer the electron to a mobile electron acceptor, that is ubiquinone. Sometimes it is pronounced as ubiquinol. Basically, ubiquinone is a ketone that is oxidized form and ubiquinol is alcohol that is reduced form. Ubiquinone takes electron from iron sulfur protein and converts into UQH2, that is ubiquinol reduced form. Till that, NADH dehydrogenase enzyme carry out this reaction. From ubiquinone, electron moves to the complex 3. Complex 3 is basically composed of 3 kinds of protein, that is cytochrome C1, iron sulfur protein and cytochrome B. Ubiquinone directly transfers the electron to cytochrome C1 to iron sulfur protein and then cytochrome B. From cytochrome B, electrons are transferred to a mobile electron carrier, that is cytochrome C. Cytochrome C transfers the electron to 4th complex, which contains 2 kinds of protein, that is cytochrome A and cytochrome A3. Cytochrome A3 at the end transfer the electron to half molecule of oxygen and transfer it into water molecule, which takes the proton from the matrix of mitochondria. Till that, we have not talked about complex 2. Complex 2 is basically not directly transfer the electron. It takes the electron from FADH2 and transfer them to FAD2 positive. These electrons are then transferred to ubiquinone, that is mobile electron acceptor, and these electrons are transferred to cytochrome 3. FADH2 is basically produced from the transferring of succinate to fumarate during Krebs cycle. All the transfer of electron from ubiquinone to complex 3 and complex 3 to cytochrome C and then complex 4 is similar as that transferred by complex 1, which takes the electron from NADH. The transferring of electron from FADH2 to ubiquinone is characterized by enzyme succinate dehydrogenase. In addition to these 4 complexes, the electron transport chain of plant mitochondria contains 5 additional enzymes marked in green. None of these additional enzymes pumps protons. Basically, some specific inhibitors, rotenone for complex 1, antimycin for complex 3, cyanide for complex 4, and salicyl hydroxamic acid for the alternative oxidase are important tools to investigate the electron transport chain of plant mitochondria. Complex 1, 3 and 4 have the ability to pump the proton from matrix to intermembranal space. Complex 1 transfers 3 proton while complex 3 transfers 4 proton and complex 4 transfers 2 proton from matrix to inner membrane of mitochondria while transferring the electron to the next electron carrier. The transfer of electron from the matrix to intermembranal space creates a proton gradient. These protons move from high concentration to low concentration through ATPase enzyme or ATPase complex. ATPase is basically a complex of F0 and F1. F0 is channel and F1 is head. When proton travel, head revolves and ATP is produced. ATP synthase is also called complex 5. The transfer of 3 protons from intermembranous space to matrix gives 1 ATP molecule. In this way, we can conclude that 1 NADH molecule gives 3 ATP molecule while 1 FADH2 molecules gives only 2 ATP. Because complex 2 takes electron from FADH2 and give to ubiquinone, it does not directly pump the electron while transfer it to ubiquinone, which then again transferred to the cytochrome complex 3 and 4. In this way, energy is produced in the inner membrane of mitochondria that is called oxidative phosphorylation or chemiosmosis. If you have not watched how NADH and FADH2 are produced in the matrix of mitochondria or in the glycolysis, you can click on the I button or link in the description. If you like this video, give a thumbs up because it motivates me to make more informative videos.